Professor Michael Tomashow is our speaker today, and although essentially all the speakers that we bring up here are fantastically distinguished, I have to say Mike is possibly the most distinguished, or in a very select group of faculty on campus, and I have a very long list of his accomplishments that I'm going to read to you because I think it's fully worthy of our attention, at least for a few minutes. Dr. Tomashow received his bachelor's and PhD degrees from UCLA, majoring in microbiology. He conducted postdoctoral research as a Damon Runyon Walter Cancer Fund Research Fellow at the University of Washington. And his first faculty position was at Washington State University, followed by his moving to MSU, where he's been since 1986. Prior to coming to MSU, Dr. Tomashow worked on the plant pathogen Agrobacterium tumefaciens, which causes the formation of crown gall tumors. He and his colleagues discovered that this bacterium transfers its DNA to the host plant and uses that transferred DNA to manipulate the plant for the bacterium's own use. These studies demonstrated that this particular bacterium is a natural genetic engineer of plants. These studies, subsequent studies by scientists at Monsanto and elsewhere, converted Agrobacterium into the primary system for producing GMO crops. So the primary system for manipulating crops. However you feel about GMO, I think this is an example of basic research coming out of the laboratory and affecting all of our lives, and so it's quite a good example of that. Upon coming to MSU, Mike received support from the Agricultural Experiment Station, which is now called AgBio Research, that enabled him to initiate a completely new line of research, some of which he'll talk about today, determining the molecular basis for plant freezing tolerance. How do plants tolerate low temperatures? How do they resist low temperatures? What biological pathways, molecular pathways in the plant allow it to do that? This support led to the establishment of a highly successful research program that has produced many important findings, including the discovery of the first freezing tolerance pathway in plants, which is called the CBF regulatory pathway. Dr. Tomashow holds the title of MSU University Distinguished Professor. He has served as president of the American Society of Plant Biologists and is director of the MSU Department of Energy Plant Research Laboratory from 2006 until this fall. Dr. Tomashow is a recipient of the Stephen Hales Prize given by the American Society of Plant Biologists, the Alexander von Humboldt Foundation Award. He's an elected fellow of the American Academy of Microbiology, the American Society of Plant Biologists, and the American Association for the Advancement of Science. And he is finally an elected member of the National Academy of Science. So it's with great pleasure that I introduce Mike to you, and I hope his presentation lives up to my build-up.
Thank you very much, Steve, for that generous introduction and the opportunity to talk a little bit about the work that we've been doing. And at the end of the talk, what I'd like to do is highlight some new efforts that are underway about establishing a center in the area of plant stress biology. Now, I realize I have 8 minutes, so here we go. All right, so on the planet now, there are about 7 billion people. In 2050, there's going to be about 9 billion people. And the question is, how are we going to feed this increasing world population? And there's actually some serious problems here, challenges. You can see this here. This is yields with maize in these black lines over here. These dots right here show what the increases in yields have been over the course of the last few years. And great progress has been made. This is mainly through traditional plant breeding. This solid line right here is where that's continuing to head. But the problem is that this dotted line is where we need to get on if we're going to feed 9 billion people. In rice over here, this progress is made and this is where things are headed, but we need to do that. Okay, so this is a major challenge. It's widely acknowledged. It's been covered in many different kinds of reports, including this one here from the Royal Society. And the idea is that we are going to have a second green revolution, and that's going to have a lot of component parts, but one of those component parts is to increase the stress tolerance of plants, the abiotic stress tolerance, that is tolerance against drought, high temperatures, flooding, and also biotic stress tolerance against pathogens and insect pests. So how is this going to be accomplished? Well, there's going to be continue, of course, plant breeding efforts, but that's not going to be enough. And what we need is additional approaches. And so the idea is that if we understand more about basic plant processes, and in particular here with plant stresses, if we understand more about that, that'll lead to new ways of improving plant stress tolerance. Okay, so this sounds nice, but the question is this just totally fanciful? Are there examples of this? So what I'd like to do is just give you a case study with the work we've been doing with freezing tolerance. Okay, so we all know that plants differ greatly in their ability to deal with freezing temperatures, right? Tomato has very little freezing tolerance. Wheat has a considerable amount of freezing tolerance. But what's important to realize is that this is not a constant property of the plants. What wheat can do that tomato cannot do is sense the dropping temperatures in the fall and activate mechanisms that lead to the enhancement of freezing tolerance, and that's called cold acclimation. This is an example of it with Arabidopsis, which is a really powerful model plant to study basic plant processes. And if you grow the plants at warm temperature and then you do a freeze-thaw cycle, they die. If you grow them at warm temperature and then put them at cool temperatures for a while, low but non-freezing temperatures, and then do the freeze-thaw, they survive. So the big question, of course, is what's occurring in response to this low temperature that leads to this enhancement of freezing tolerance. So I have my PhD in microbiology. I got interested in this and I wanted to work on it, and fortunately a job came open here at MSU and with support from the Ag Experiment Station, we initiated a new line of investigation. And to make the story short, it turns out we had a number of hypotheses. We tested those, and that led to the discovery of this first freezing tolerance pathway in plants. In fact, it's still the best understood pathway in plants. And basically, it's simply this, is that the plants sense low temperature, and then it turns on these genes called the CBF genes. They're sort of like master switch genes. Those genes, when they're turned on, they produce these regulatory proteins that then turn on a whole bunch of other genes. And those act together to lead to this enhancement of freezing tolerance. And let me just show you this a little bit more graphically in this next slide here. So here we're just looking at the expression of a few genes in the plants and asking whether or not they're on or they're off. And what you see here, this is a normal plant at warm temperatures. The CBF genes are off, and the targets of the CBF genes, they're off. But you can create transgenic plants where you switch those master regulators on at warm temperature. And that's an example here in these transgenic plants. And you can see here you have now all these genes on, the targets are on at warm temperature. Okay, so now what happens with freezing tolerance of those plants? Well, what you can see is now, even at warm temperature, these plants are freezing tolerant. In addition, they are tolerant to drought stress as well. I don't have time to go into that cross protection. Okay, so this is with Arabidopsis, a model organism in a lab setting. Can you take this to the field with a crop species? And I'm just going to quickly describe experiments that were done at Arborgen. So they were interested in developing a eucalyptus for a bioenergy crop. The problem is they are from the tropical, and they have very little freezing tolerance. And so you can grow them in this part of Florida, but you can't grow them up in the Gulf Coast. Okay, the occasional frost there it wipes them out. So they wanted to put CBF genes in. MSU held the patents for the CBF genes. They come to MSU, they sign the right papers, and they do the experiments. And what they found was this. Pretty exciting result. These blocks of trees over here are the normal trees that are not expressing these CBF genes. Here are these live ones, are the ones that are expressing CBF. So that's pretty exciting. Unfortunately, it didn't make it to a commercial product, and the reason is these are GMO plants. The regulatory hurdles to get that out in the field is really difficult. In addition, environmental groups were a bit worried about this, and basically the hurdles were too high. They now grow the plants down in Brazil. Okay, but for the purpose today is that it shows that indeed you can go from a hypothesis in the lab and go to the field and have a crop species that's improved in a complex agronomic trait. Okay, so I'll just make one other remark, and that is these are GMO, but I know the number of you are aware that there's this new kind of gene editing technology called CRISPR kind of technology that one can potentially do these things and get around being a GMO plant. Okay, so indeed this is not fanciful. Knowing more about basic plant biology lead to new ways of improving stress tolerance. And in that context, I then just like to end on this last slide. It'll just take me a couple minutes. A number of us are excited about establishing a Center for Plant Stress Research. I hope you're convinced this is an important area of research. Really, this came out of the ACF initiative, and now the Global Impact Initiative. And as we know, the idea is, well, okay, MSU is strong in its research. But the challenge to the faculty was, well, how are we going to take it to the next level? What kinds of things to take that to the next level? Well, one of the areas of strength on campus are the plant sciences. And within the plant sciences, one of the strengths are plant stress biology research. And so 4 of us got together here. Well, actually more, but we spearheaded this. Myself, Brad Day, Shenyang He, Greg Howe. They are internationally known for their research, highly regarded. Shenyang was elected to the National Academy last year. And what we decided to do was put forward a proposal that had 2 components. One was a cluster hire, right, to bring on additional faculty that could strengthen areas of research in plant stress biology and broaden and deepen that. But in addition, we thought that to take full advantage of this, it would really be a powerful situation if we could also have a center where we brought the people from campus and we brought new people from off campus together to conduct collaborative research to address really fundamental questions in plant biology, in stress biology. So we envisioned then the formation of this Plant Stress Research Center. We don't quite yet know what the name will be, but we envision it to become an internationally recognized center of excellence. We have such entities on campus. For example, in the plant sciences, the Plant Research Lab. It's internationally known, but we have really strength here. We should have more of these focal areas, high-profile entities that people can point to. In addition, this center, we envision it to promote excellence in research and training, and also, very importantly here, is to position ourselves to be highly competitive and to win future awards in grants that are going to be directed in this area. And finally, to contribute to attracting exceptional faculty. You know, it's one thing to come into a university, into the departments, which is very good, but if you come in and you are part of a new effort, there's an added draw to that kind of position. So we would hope that this would also assist in that regard. So we put forward this proposal. It met with enthusiasm in the VP's office, in College of Natural Sciences and Ag Bioresearch, and also in CNR. I hope it has some support in the President's office and the Provost's office.
If you haven't gotten the money. Provost Hewitt was the money keeper.
All right. So where we're at right now is we've been approved for the cluster hire. We are putting the pieces together to fill in this picture for the center, and hopefully within the not too distant future somebody else will be standing up here and tell you about the accomplishments that come out of this new center. I mean, again, thank you very much for
questions for Mike. The board yesterday we went we did sort of a field trip over, and Brad Day was very gracious to take us around and some of the spaces and talk a bit and see students and faculty at work.
Yes.
And hearing about your new announcement of getting a couple of $5 million grants, by the way, as Christmas presents for you all last year. But questions,
They're
comments?
excellent.
Questions, comments for Mike?
Well, they teed us up for you yesterday.
Yeah.
But I can see why we're winning these grants. When we tour the facilities and see how first class they are, and then we end up interacting with everyone over there. Because when we discover that they're not giving the grant based upon presentation, they're giving the grants based upon touchy-feely. And that's why Michigan State's going to, they're going to meet individuals like you and the rest of the individuals over there, and we can see we're really making a difference. So we're very thankful to be teed up yesterday, and we continue to be impressed with your presentation.
Thank you so much.
Questions, comments, anyone?
I have one question.
Yeah.
Coming off from the Paris climate talks and well, there was a lot about climate smart agriculture, which is not really defined. And obviously when you're looking at extreme weather events, drought tolerance, heat, frost, what do you see coming up? What do you see in climate smart agriculture in the next 5 to 10 years? What kinds of research and practices?
All the things that I was talking about here today are, we need to improve the stress tolerance of these plants. Okay, that's a given. It's widely acknowledged. Okay, all of those, that problem is just even made more uncertain with climate uncertainty. So the part that I can speak to is really the science side of it. I really can't speak at all to the policy side in that business. But it's absolutely clear that if we're going to meet the demands of feeding 9 billion people, if we're going to have food security, high quality food, et cetera, that we are going to have to do a number of things, one of which is improve our understanding of these mechanisms so that we can come up with novel ways of improving the stress tolerance of plants. And again, like I say, the climate uncertainty is only going to exacerbate that and make it even more important.
Could you, we talked a bit yesterday about GMOs and what it meant, and you commented that the eucalyptus was project moved to Brazil rather than being in Florida, but then you mentioned a new technique. What does that mean in terms of being able to move some of these ideas from the laboratory into helping people feed the world?
Right. Well, so this new technology, instead of taking, with agrobacterium, as Steve was pointing out, we can take genes and put them into plants, and that's incredibly powerful. That will remain powerful in an experimental setting, but there are concerns about GMOs, okay? So the wonderful thing is that this new technology of CRISPR, you can actually go in, edit the sequence in the genome, and hopefully get these same kinds of outputs, but it's not considered, there's a number of decision-making processes that are underway. It looks like in the United States it will not be considered to be GMO. It's just mutation within the genome, which is what plant breeders use all the time, okay? And you select on that. So this will be just more designed, but it will not fall under a GMO category. It's not quite clear what's going to go on in Europe at this point. But the point is that we need to develop ways of improving various traits in plants. And the fortunate thing is that this kind of technology should be able to be applied. So it's very promising future.
And new kinds of technologies will also evolve in the future, but it still requires the work that you and your colleagues are doing to understand gene expression, gene mechanism, everything about it. It doesn't matter what the technique is as much as
right,
you have to have all the fundamentals.
Right. That part, the GMO part, is bringing it to the field. This CRISPR is bringing it to the field. In terms of the basic knowledge, though, doing the work in the lab is those are the things that we can use the current technology and this new technology.
Okay. Well, it's terrific to have you be one of our beacons in building plant science here over a very long period of time, and it's great that with your you and your colleagues, in terms of the enthusiasm, the passion for this work, that the new center, I think, is going to be extraordinary moving forward. So thank you.
Thank you.
Thank you.